Assalamualaikum and hello everyone. My name is Nur Azina binti Hassan and I will give further information about my final year project titled as Microorganism associated with Pangasianodon hypophthalmus cultivation. Fish is a vital source of food for people other than meat. Fish are easy to digest, contains a high protein with a low fat food that comes out with a range of health benefits. Fish also have a high nutritional value, including omega-3 fatty acids. When it comes to food, there is a chance that the food can be contaminated and becomes a liability to the human when eaten. Fish is one of the major protein that gets easily contaminated by bacteria due to a high water content and ambient temperature. It is also due to excessive bacteria and another pollutant in the water and cause them to get infected. Pangasianodon hypophthalmus is a freshwater fish known as Iridescent Shark, silver catfish, or in Malaysia we call it as Patin. Based on many studies conducted by many researchers, it is proven that spoilage in fish is due to microorganisms that infect them. Fish are easily contaminated, especially when not stored at the correct temperature in the fridge. It is essential to know that once the food is spoiled or contaminated with pathogenic bacteria, it becomes toxic to humans that consume the food. When the food enters the human, it causes many foodborne diseases, including dysentery, typhoid, meningitis, and severe food poisoning. Management on water quality and cultivation type influence the degree of contaminated fish. Okay, diagram below shows the newspaper headline on food poisoning caused by seafood, especially fish that was contaminated by pathogenic bacteria such as E. coli and various Vibrio species. Fish are more susceptible to bacterial invasion from the external environment during normal instance and spoilage. The possible contamination source for these bacteria have been a poor hygienic fish handling practice and unsafe fish storage conditions. Thus, it can be identified whether they affect the storage life and quality of the fishery product. Detailed information on the bacterial load and types of bacteria associated with healthy fish organ is needed to provide a predictive capability for possible outbreaks. Apart from that, it is also an opportunity to design preventive management action. This study also provides an extensive literature review for the importance of foodborne disease and freshwater fish economics prospect, particularly in Pangasianodon hypophthalmus. The objective of this study are to collect data on the cultivation type, economic value, and microorganism associated with P. hypophthalmus disease. Next is to discuss the treatments of P. hypophthalmus cultivation due to the microorganism that cause the disease. Lastly, to suggest alternative cultivation and biological control on P. hypophthalmus cultivation. P. hypophthalmus from family Pangasiidae is a freshwater fish that migrates from upriver to generate offspring during the flood season and come back to downstream to find nurturing habitats when the river water backs to normal. P. hypophthalmus is a native in the Mekong River and have been introduced to many Asian countries including Malaysia, China, Philippines, Singapore, Taiwan, Bangladesh, and Nepal. P. hypophthalmus has a lateral and elongated body shape. They also have dark gray or black colored fins. They have 6 dorsal fins ray that were branched. They usually have developed gill rakers. Small fish has black colored stripes along and below the lateral line, while large adult fish have a generally uniformly gray stripes. They also have dark stripes on the middle of the anal fin and in each of the caudal lobes. P. hypophthalmus need protein for their growth and development but in a low quantity. They do not need a large volume of protein and the excess protein will be excreted as ammonia. Fat and carbohydrates are needed to provide the P. hypophthalmus with energy to swim. Minerals such as phosphate and calcium are needed for the bones building block. Vitamin C is needed by P. hypophthalmus as a catalyst for chemical reactions. The appropriate range of water temperature for P. hypophthalmus production was from 28.2 to 31.7 degrees Celsius. The drastic pH fluctuations will result in P. hypophthalmus stress. The appropriate range of water pH for P. hypophthalmus production was 6.8 to 7.6. In aquaculture, probiotics have been used to increase the growth of P. hypophthalmus to enhance digestibility and treatment of nitrogenous waste. Freshwater fish is a biological agent for mosquito control. Fish prey on the mosquito larvae to keep mosquito populations under control. Freshwater fish commonly eat on mosquito larvae, which will reduce the number of mosquito release. P. hypophthalmus is considered as a healthy diet food based on the nutrients in their meat. The fish has a high protein levels, minerals such as calcium, essentials, fatty acid, and vitamin A. Other than consumed as food, P. hypophthalmus play an essential role in economics. Exploitation of P. hypophthalmus contributes to many countries' revenues, including Vietnam, India, and Cambodia. The table below shows the area and figures projected within aquaculture industry zone in 2009 in Malaysia. Many bacterial species can be found in fish, including E. coli, Enterobacteria, Klebsiella, and Serratia. The significant genera that are found in Pangasianodon hypophthalmus include Vibrio, Enterobacteria, Aeromonas, and Serratia. The research also detected three types of Staphylococcus, which are S. coni, S. lentus, and S. intermediates. Foodborne pathogens take advantage of weak immune systems. Infant and young children, pregnant mothers, the elderly, and the immunocompromised are particularly at risk of contracting and dying from common food-related illness. Vibrio species accounted for 38% of the outbreak associated with seafood and 54% of the disease. The diagrams below shows the bacterial flora identified in fresh and spot freshwater fish. Based on the literature, it is essential to note that if the food is poisoned or tainted with pathogenic bacteria, it becomes harmful for people who eat the food. It is important to know and list the microorganism involved in freshwater fish, so proper treatment can be applied to the cultivation system. The literatures also discuss the cultivation types, economic value, biological controls, and water quality of freshwater farming. So, the collective information are vital to understand the information and future prospect of P. hypophthalmus in Malaysia. For recommendation, the idea is to create policies and regulation for fish cultivation in order to control water quality. By controlling water quality, maybe the number of bacteria in the fish can be reduced. Another idea is to study and report the migration of larval drift and fish fry. A better understanding of the process of spawning and its natural circumstance will contribute a great deal to adequate protection. Last but not least, is to monitor the abiotic and biotic parameters must be conducted regularly using the Internet of Things IoT technology. Please visit my Facebook for further information about this research. Thank you very much for your time and Assalamualaikum.